It is now about 9:30, I think, in the morning for Gerardo Corzo Burguete. He is a professor at the Institute of Biotechnology at the National Autonomous University of Mexico. And Gerardo is a very prolific researcher. He's worked on arachnid venoms, on reptile venoms. He has looked at the bioactivities of venom toxins. He's looked at antimicrobial peptides, and he's also studied antivenoms. And it's this latter topic that he will talk to us about today. So Gerardo, thank you very much for joining us at the EUVEN seminar series. The digital platform is yours.
Okay, thank you very much for inviting me. And I want to say hello and thank you to my friends from Turkey, Hakan, Ferhan, Figen. Thank you very much. So I will share my screen, and the presentation is not here. I don't know where is it.
Take your time.
Yes. Okay, it's here.
Yep, we can see that.
So I want to talk a little bit about what we have been doing concerning the expression of neurotoxins from scorpions and elapids. And thank you very much for giving us this opportunity. As you will see, there are many co-authors or authors. Most of them are students and technicians, researchers now at the laboratory, or they had been here. So what is interesting in here, I will start passing the diapos. As you see, scorpion and elapids of medical importance, there are some genus. They are very interesting and they are dangerous for people, most on the tropical areas. You will see in white. I don't know how to. I don't know. You will see my cursor. This is true. So
yeah, we can see your cursor.
Okay. In black, this is the genus from scorpions. For example, in the north of America, we have the genus Centuroides, in the south Titius, Androctonus in North Africa, and Middle East Lirius and Androctonus, and others in South Africa, Paragutus. And the genus of the medical importance and the elapids of medical importance are Micrurus in America, Naja, Dendroaspis, Ophiophagus, Oxyranus in Africa and Asia and Australia. So we are talking a little bit about this genus, and what we have been doing concerning expression of neurotoxins that we use for immunization in rabbits and horses, and see how they can, even a single toxin used as immunogen could produce antibodies for neutralizing the same toxin and the venom of scorpions or elapids. No? These are some of the species that we have been working with. Titius from South America, Centuroides, all these are elapids, and this is a coral snake from America. This is Botros, they call it now Yaca here in the tropical areas of Central America. Because of the time, we are not going to talk about viperids, but we will talk about elapids. To introduce you, you may know all about this, no? What we are thinking in a simplistic manner is that the toxins from scorpions that affect mammalians and humans, of course, there are one group of toxic proteins or neurotoxins, the ones that affect the sodium channel, the voltage-gated sodium channel. They are separated by alpha or beta neurotoxins depending on the mode of action. And in elapid neurotoxins, there are two groups of toxic proteins. Mainly, they are the three-finger neurotoxins. There are also small ones, like the neurotoxins from scorpions and phospholipases. No, the differences, they affect, as I said, voltage-gated sodium channels, the scorpion neurotoxins, And the elapid neurotoxins, they affect acetylcholine receptors. In a simplistic manner, what we are trying to do is to express one of these neurotoxins and to see if they can be used as immunogens to get antibodies or help to increase the number of antibodies in antivenoms that neutralize these toxic proteins for mammals. The commercial antibodies are diverse. They are based in antibodies mainly from horses, from antibodies from horses. Mexico has mainly three pharmaceuticals. Two of them are private ones. This is Bioclon, Beteria, and this is a public one. And other countries, of course, they have their own antivenoms, and most of them, because of the regionalization or zonification of these animals, or these poisonous animals that affect some of these countries. The antivenom pipeline is just taking or they are based on natural venoms. They are obtained by extraction, lyophilized, and they are immunized in horses. And after several months, the antigens inoculated into the horses generate antibodies, they maturate, and then they are ready for processing, in a lyophilized or in a soluble form. So one alternative could be recombinant neurotoxins to be as antigens to immunize horses. And again, the difference between these two, we are working here with neurotoxins that affect directly to mammalians. And here we have neurotoxins, and we have other proteins. They are a specificity, or they have specificity for insects or for other receptors that are not perhaps important for medical issues. So this presentation, I will divide it between the proof of concept, the first research that we do with the scorpion venoms, and the current work that we are doing for experimental antivenom for North Africa and Middle East. This is in collaboration with Dr. Figen in Turkey, and something that we have done with neurotoxins from elapids. So the first one, as I mentioned, the scorpion venoms, when the people is stung by these animals, they induce a massive release of neurotransmitters. They cause sweating, nausea, vomiting, hypersalivation, and they can lead to death. So in Mexico, we have mostly 3,000 people that are stung by scorpions in a year. So the high is very high rate of scorpion accidents in here. In Mexico, we have the booted genus Centuroides, Centuroides diffusus, Noxius, Centuroides limpidus. There are several species of Centuroides, and they are along the Pacific coast. In here, in this area, from the Atlantic, the Mexican Gulf, there are also scorpions. There are other genus, but they are not of medical importance. So at first, we start looking for neurotoxins that are active on mammals on this species. And of course, there is a lot of research that we have been doing with these scorpions since 1970s and all that. Here is the protein fractions separation by HPLC, reverse phase HPLC of this scorpion. There is Centuroides diffusus. And as you can see, there are several protein fractions. We know which fractions they affect potassium ion channels, which affect sodium ion channels. Some of them, like this high one, is insecticidal, but this one is mostly the most representative toxin that affect mammals, mice. No? If you take these toxins from the venom is not as toxic as containing this protein. No? And the LD50 is very low. And there are also small toxins that affect mammalians, but they are in such a small amount that the effect is not significant. Huh? And this toxin, it comes in almost 2.6% of the soluble venom. So this is the main target neurotoxin on this venom to be neutralized. And family of neurotoxins, they are quite identical, no? In Centuroides, in the genus Centuroides, for example, from Centuroides noxious, we have this protein, these neurotoxins. From Centuroides limpidus, we have this with small changes in amino acids. So the identity is around 85% to 100%. So most of them, there are one neurotoxins that are similar to the other ones that affect mammalians. So here we use this toxin CCS2 to be expressed recombinantly using a expression system that is, we use this expression system since 2007, is taking the protein in a plasmid that is PQ30, and this protein, it has a N-terminal fusion, an open reading frame, a histidine tags, and a site for enzymatic cleavage in case we would like to release the mature neurotoxins from the recombinant protein. No? And this protein has several steps until their purification. First, we get it from inclusion bodies. We purify by histidines resin, by nickel resins. We reduce to once we once we have the inclusion bodies in the cell and we purify them, when we solubilize them, we think that these proteins get oxidized. So it presents several isoforms with the same molecular weight, as you will see in the next slide. And then we had to reduce it. We had to fold it in vitro, purify again. We can cut it by the enzymatic cleavage and purify it again. So in this case, we'll have a really mature protein similar to the native toxin. One important thing to consider here that not many articles or not many people, researchers that work on this kind of topic of expression of neurotoxins is the amount of cysteines that the toxins has. For example, here has 8 cysteines and they form 4 disulfide bridges. So mathematically, 4 disulfide bridges could be formed 105 possible isoforms, no? Just not taking this cysteine bridge. It could be this one with this one, this one with this one. So the combinations of the possible combinations are 105. Huh? But nature just choose one that is the toxic one. No? This is quite interesting because expression of proteins of this type has to be, be sure they are toxic and they have the right structure. Huh? So this is the cell extract of a cell that has the inclusion bodies, and we can perform a SDS patch gel. In here, for example, this is protein markers. The number 2 are the complete cells, proteins in the cells. Line number 3 is a recombinant protein after affinity chromatography by nickel. And number 5 is supernatant. So most of the neurotoxin go to inclusion bodies, and it make this kind of dimers or tetramers and all that. Even we have reduced with mercaptophenol and we use SDS, we always see this kind of dimers and all that. But there are dimers that are not covalent bonded, no? Because if we take this line or this protein in here, and we perform also an HPLC by reverse phase, what we can see is several protein fractions that have the same molecular weight. This is the molecular weight that we are expecting by the primary structure of the protein. But what is interesting, none of these fractions are toxic. So what we can, how to say about this is that these proteins, they are oxidized, but in multiple isoforms connecting different cysteines, like a scramble protein. No? But if we take all these proteins and we reduce it with DTT, in this slide, we have also an HPLC of the multiple isoforms, and we reduce it by DTT, we break all the disulfide bridges, and we gain 8 protons. So the molecular weight is, in this case, 9,400.6 daltons. And there is one single protein fraction that has this protein without any disulfide bridges. So if we take this one and we reduce it, we oxidize it, here we oxidize it using a par redox. In this case, we use glutathione reducing oxidize it. We go from the unfolding protein to a folding protein, and we set in more controlled way under a pH 8, some concentration of chaotropic agents, salts, trees, whatever, but with a par redox. So we got this protein, and this one is quite toxic, similar to the native one. So by folding the proteins in a controlled way, we can get what we consider is the correct structure of the protein. So this protein, if we cut by factor XA, this enterokinase, kinase, we can cut here and release the protein, the fusion proteins that comes in here, and take the recombinant protein or the mature protein in this form. The secondary structure of the native toxin, CCS2, with the recombinant and with the N-terminal fusion protein, is quite similar. They have typically the alpha beta fold of the scorpion neurotoxins. And of course, in here, we cannot see in detail if there is any right disulfide bridges as the native one. So that's the reason in 2012, with the group of Federico del Rio, we compare the recombinant toxin CCS2 with the native one. This is one proton spectrum of the magnetic resonance, and here is the NOSI spectra. And what we can see here is the vibration of the hydrogen of the carbon alpha of the cysteines. And we make a comparison with the native toxins, and by this result, we observe that the cysteines 1 and 8 see the cysteines 2 and 5, 3 and 6, and 4 and 7 were connected, and it has the same structure. No? So by this way, we understand that folding the neurotoxin, this CCS2, in a controlled manner, we can get the same structure of the native toxin. No? So taking in account this, we perform immunization of rabbits using the recombinant toxins without the fusion protein, the native toxins, and the whole recombinant protein with the fusion protein. So we got titers and we got antibodies, and we observed that was the antibodies obtained by the immunization of rabbits, or the rabbit antibodies, they can neutralize the toxin, other toxins that are similar to CCS2, and we can neutralize the venom, the total venom of the Centroides suffusus. So this was our proof of concept that recombinant peptides, even with the fusion protein or without the fusion protein, are immunogenic and produce neutralizing antibodies at this one. No, so this was the first work that we did concerning expression of proteins, and then we work with Dr. Figen in Turkey. That is the next part of this presentation. But first, what I want to show to you, you may know, is the toxins or the neurotoxins from the genus from different regions are quite different. For example, these are neurotoxins from Centuroides from North America. And the identity is quite close, no? From 86% to 100%, taking reference to the CCS2. But if we go to the neurotoxins that affect mammalians from South America, it drops to less than 50% of identity. No? But if we go to the same from neurotoxins from North Africa or Middle East, it drops almost 70 or 60, 70% difference. So the antibodies that we can get by using this antigen, CCS2, of course, they will not neutralize these other toxins, no? And also the neurotoxins that are just specific for insects, they also quite different to the neurotoxins that affect mammalians, no? So it is interesting because after the slides that will show about the neurotoxin from North Africa, you will see the alignment or the identity of elapid three-finger toxins, even from America, Asia, Australia, and Africa. The identity is more closer than these ones, no? So in one way, perhaps scorpions they have been evolved more than elapids. No? So in North Africa and Middle East, we had also dangerous scorpions. They are larger in size than the scorpions in America. And most of these important scorpions are Androctonus australis, Androctonus mauritanicus, Crassicauda from Turkey, Saudi Arabia, Liurus, Androctonus, Parabutus from South Africa. So, but in here, what is important is only one or two neurotoxins from these venoms are quite toxic, similar to the neurotoxins from North America. Or as the first slide that I showed to you, there is mainly one component that is toxic, and if you tackle this neurotoxin, you may have an important gain in antivenom neutralization, no? Just using one neurotoxin. And of course, the amount of neurotoxins in this venom is quite perhaps 3 to 5% of the total venom. So it's important, no, for the amount of venom that they inject to humans when they're stung. So to perform this work, we know that by Dr. Figen's laboratory, the Androctonus crassicauda has main toxins in their venom. They call it ACRA1, ACRA2, ACRA3, ACRA4. And this ACRA that was recently published by an Iranian group, I think, or I don't remember. And the main toxins from Androctonus australis are these three. They are quite toxic. These are the sequences. And that we observe is the most toxic fraction or most toxic protein from crassicauda has this amino acid sequence. And the amino acid sequence of Androctonus australis is quite different in this region. So, but this sequence is more close to other toxic compounds from other species from North Africa. So we performed an alignment and we made a consensus toxins from North Africa. That is this one. When there were indefinitions, we take the amino acid that was more representative for the toxic proteins, and we got these neurotoxins that was produced by reverse transcription. I mean, this is a new toxin, this is a novotoxin that is not in nature, no? This is the work of the students, Samuel Cardoso. And what is interesting, this part that is different in the amino acid sequence of the neurotoxins from other scorpions, they also have presence in other toxins like Androctonus australis. So in North Africa and Middle East, scorpions has two main neurotoxins. One is close or identical to ACRA4. Sorry. If we take ACRA4 as 100%, and we take these neurotoxins that is quite different to the others, to the consensus toxins, it drops to 50% identity, and vice versa. If we take 100% of the consensus toxins to others, they are quite different. So in here, we express these two neurotoxins to see which one is the most immunogenic, and if by getting antibodies from one of these toxins, we can neutralize the whole venom. So we use the same protocol for protein expression, same plasmid or similar plasmid, PQ30, the promoter of the phase PT5, a histidine tag. And in here is no CCS2, but this is the cassette for the consensus toxin for North Africa and ACRA4, and the same steps. No? Here we can obtain a well-folded toxin, but we found a protein fraction that was toxic to mice. For example, from here there were 4 to 5 protein fractions, and we purified this one that was toxic to mice to be used for immunization of rabbits. In here, also, we get these neurotoxins, recombinant neurotoxins that were oxidized. They were not folded as the CCS2 toxin, as I showed in the first part of the exposition, but we got the protein fraction that was the most toxic to mice. And taking these two neurotoxins, recombinant neurotoxins, we immunize rabbits, and we perform some recognition by ELISA. And as you see in here, this is a recognition of antibodies to the recombinant ACRA4. And of course, there is a lot of recognition because we immunize rabbits with this neurotoxin. No? And in here, it was the recognition to the venom of the crassicauda, Buctus occitanus, Lierus quinquestratus. No? It was less recognition. And on the other hand, the antibodies that were raised by immunizing rabbits with these consensus toxins, also they have a lot of recognition by the recombinant toxin, but it has a little bit more recognition for the Androctonus australis, the native one, yes. and it's quite similar to the native one, and recognition to also venoms of Crassicauda, Occitanus, Androctylus actralis, and Lirius quinquestratus. No? So we can observe that the immunization of rabbits and the antibodies obtained in them, they can recognize neurotoxins, and they can recognize neurotoxins within the venoms, no? And also native neurotoxins. So with this information, we perform neutralization experiments, and this is a big table. But what is showing is the venom or toxin of Androctonus australis, Crassicauda mauritanicus, and these venoms. This is the LD50 reported by these venoms. We use 3 times this amount to challenge mice. And for example, by injecting 3.5 micrograms of Androctonus hector to the control, no one survived, no? But if we use the same amount and we challenge with 10 milligrams of antibody from rabbits, all mice survive, no? It's letting us that the antibodies that were raised using the consensus toxin for North Africa, 10 milligrams can neutralize the venom and this. But for example, from Androctonus crassicauda, it's the same. We inject 19.2 micrograms to mice. This is the control. But by taking just 10 milligrams of anti-ACRA4 antibodies, they non-neutralize the venom, and also they non-neutralize with another toxin this venom. We have to use a combination of these two to neutralize the venom. No? So in the next slide, this is a zoom of this information that I am showing you. And what is interesting is some venoms could be neutralized by the single antibodies raised by one antigen, but other venoms perhaps are more complex, or they can have a different composition of these two types of neurotoxins. You remember CCS2 and ACRA4, they differ in 4 or 5 important amino acids, and this can be important for toxicity. No? And the combination of these two, they can neutralize the venom. So, of course, in a simplistic manner, we think anti-scorpion venoms contain one neurotoxin that are toxic, but they can have 2 or 3, and they present different antigenic groups. So this is the information that we are showing to you. No? Going to the last part, so recombinant elapid neurotoxins, the elapid venoms, when people is bitten by these snakes, they contain neurotoxins that paralyze muscles in the thoracic cavity, mainly the diaphragm. So respiratory paralysis occurs, and people die in few hours if not treated with antivenom for elapids. No? So these are some of the lapids that we are working with, or we have been working with. This is the HPLC of different lapid venoms. No? These are coral snakes from South America. These are coral snake from Africa, and this is Nahacautia from Asia. No? And what is interesting is in this rectangle, what we are showing is most of these fractions are 3-finger toxins. They are 3-finger toxins, and the ones that have larger attention times, there are phospholipases. So South American coral snakes, they contain more 3-finger toxins than phospholipases, and Sí, NAHA contains also most neurotoxins than phospholipases, but NAHA has both, no, neurotoxins and phospholipases. And it's important because there is a report from Bruno Lomonte some few years ago saying that in America, the North American coral snakes, they had more phospholipases than three-finger toxins, and the South American coral snakes, they had more neurotoxins of three-finger type than phospholipases. So every region, they have similar, different composition in the neurotoxins and the phospholipases. And of course, it depends on the niche, on the surroundings, and the prey, and the defense that they should have for surviving. No? But what is interesting is the three-finger toxins, they have these kind of structures. No? There are two types, the short three-finger toxins and the long three-finger toxins. The long ones has an extra disulfide bridges. They have also 4 disulfide bridges, and long has 5 disulfide bridges. And similar to the scorpion toxins, folding a recombinant toxins or 4 disulfide bridges, it makes 105 possibilities, no? Mathematically. And long alpha neurotoxins, of course, they will have more probabilities to make more isophores. No? But what is interesting, as I mentioned before, is the neurotoxins. For example, this is from America. These are from Africa. These are from Asia. This is a coral snake from the oceans, a marine coral snake. And even they are from different continents, they share at least 70% identity. No? So the evolution of the short-chain alpha neurotoxin has not been so diverse of the evolution of the neurotoxins from scorpions. No? Neurotoxin from scorpions, we have that affect in a alpha way, or beta neurotoxin that affect in another way. But for the short alpha neurotoxins from elapids, they just block acetylcholine receptors, and there are not electrophysiologically differences between short neurotoxins from Africa or America. No? So in here also, taking account this information, we produce a consensus neurotoxins also. It's the novotoxins. We use reverse translation for making this, just making oligos, putting all together, and making neurotoxins. And again, we use the same system that we previously showed you in the PQ30 plasmid, E. coli, and this is the expression of the proteins. No, in here we have cells, protein cells. This is supernata, and this, of course, what is interesting here is sorry, this is in the cells, in the whole cells, but most of this protein was soluble and was taken from the cytoplasm of the cell. It was already folded, no? And here is the purification of the protein, and this protein fraction represents the consensus neurotoxins. This is the L50 that is close to the lethal dose of most toxic short alpha neurotoxins from elapids. Secondary structure is also similar, and they're active on acetylcholine receptors. No? By using 5 micromolar, they block the ion current of sodium, calcium, potassium through these receptors. No? So this protein, we produce it in a massive form. This is the protein in a small reactor, and we immunize horses. We have the opportunity to work with Professor Dr. Alagon, who provides us horses. No, this is the blood of horses after almost 5 months of immunization. And this is the evolution of the titers. No, you see, fueron 3 horses. This is the evolution of titers. Then we have as a booster of proteins of immunization. But what is interesting, these antibodies that were raised against only this neurotoxin, alpha neurotoxin, they can neutralize neurotoxins from America, or 3-finger toxin from America, and also from I think this is from Naha, from Cobra Real, Naha Age. No? And also they can neutralize the antibodies different coral venoms from America, from Africa, and even from marine elapids. No? Of course, there are some venoms that were not neutralized, and we think that this may be because some venoms contains different proportions of neurotoxins and phospholipases. No? But what is interesting is one single protein can be used as antigen to neutralize several venoms. No? Of course, in here we are not taking account the long alpha neurotoxins from elapids, and we didn't perform now how they recognize these long neurotoxins, and it's an interesting work that we are doing by expressing long alpha neurotoxins from elapids. No? But what is interesting is suggesting that one protein will be enough for getting enough antibodies for neutralize even pure neurotoxins or venoms. No? So, in conclusion, scorpion well-folded recombinant neurotoxins are suitable to be used as immunogens to raise neutralizing antibodies and to formulate commercial antivenoms or to complement them. There is a work that we did 2 years ago. For example, we know, I will go back a little bit. For example, these are venoms from North America. They have a lot of phospholipases, no? And so if we use commercial antivenoms that were raised against these ones, and we introduce antibodies raised with these antibodies with this antigen, we can neutralize neurotoxins also from or venoms from all the continent. No? So we can complement antivenoms by formulating antivenoms with one polyvalent antibody. No? A mixture of selected recombinant neurotoxins from venom of the gene androgynous produce neutralizing antibodies. That means sometimes it's necessary to have 1 or 2 or even 3 recombinant antigens to formulate one antivenom. No? And recombinant immunogens could replace natural ones. Some advantages of that is, as you see in the production of recombinant proteins, are the higher levels of reproducibility, is independent from natural sources. There is a large antibody response only to toxic proteins relevant to the venom. When you immunize an animal, a rabbit or a horse, with the whole venom, there are some proteins that are not relevant to the toxicity. No? But even though antibodies will be raised against not relevant toxins, no? And also there is the term of immunodominance. No? There are some proteins, large proteins, that perhaps they are not relevant to the toxicity, but they will take more of the attention of the immune system of the animal. No? So we can, by formulating and producing recombinant neurotoxins, we can select and we can direct to have more antibodies against the toxic compounds. Of course, no all is relevant with this information. Some disadvantages is the applications according to the complexity of the venom. No, for example, biperids are more complex than elapids. Elapids are more complex than scorpions. No, so also we haven't taken in account the synergy. No, there are, for example, those proteins that have low toxicity for mammalians, but when they are combined, they have some synergistic effect that make them more toxic than the single one of these. And other disadvantages are the incorrect folding that generates low affinity antibodies. No, they could happen. No, if we have a recombinant neurotoxin that is not well folded, that present other epitopes that are different to the native one, the antibodies that are producing are not effective as the antibodies that are produced by the native toxins. And also what is interesting is even some people said that folding is already solved, contrary what Affinsen or Levinthal said. The correct protein folding of sting-rich proteins has to be solved yet. No, there are several neurotoxins in venoms that contain 4 or 5 disulfide bridges, and if we can produce it synthetically by chemically or recombinantly, we have to take in account the folding of these proteins. No, finally, this is the picture of the students here in Mexico, the laboratory. They all have been working with expression of proteins or doing other kind of work, like working on biperids, on assassin bugs, on frogs, on antimicrobials, enzymes, whatever we find interesting. No, but thank you very much. That will be the whole presentation.
Thank you very much, Gerardo. Very interesting talk. A lot of technical details.